let's move along here and jump into our second, or actually our third, because we did touch. But let's get into taste, gustation. Okay, there's a couple different, not a couple different, but there's several parallels between olfaction and. Oh, sorry guys. It's great that you can hang on every word, but sometimes it helps to see pictures. All right. I apologize. Okay, as that's warming up and coming up online. So, like with smell, with olfaction, all right, the molecules that triggered that sense, we call those odorants, okay? Well, in gustation, we call those molecules tastants, okay? And there's specific molecules that when they interact with your receptor there, in this case, we're dealing again with chemoreceptors. All right, remember chemoreceptors are dissolved chemicals in a fluid solution. All right, so these chemoreceptors are going to be found in our taste buds. And so we're going to talk about, all right, some of the locations of the taste buds because they're on your tongue. Nowhere else in your mouth, but on your tongue. Okay, so your tongue has these bumps all over it, all right, mainly on the top part and on the sides and the front, all right, and we call those papillae. Okay, so think of these papillae as bumps. Now, some of these bumps, these papillae have taste buds in them, and some of them do not. All right, the first one listed here, the filiform ones, all right, these, all right, have no taste buds, but their job is to help move the food around or manipulate the food as you're chewing. Get rid of that. All right, so think of it like if you've ever played soccer or any type of sport that you play on a field and that you need to get traction. If you have to wear cleats, play golf, they wear spikes. If you run track, they have special shoes that have spikes on them. At least the older ones did when they ran on ash. But that's what the papillae are, all right, specifically the filiform. All right, there are these bumps on your tongue that kind of give your tongue traction. So when it's moving the food around as you're chewing, all right, your tongue, you don't even think about it. I don't even think about it most of the time when I'm chewing. But your tongue is subconsciously just moving food against your teeth, especially your molars as you're mashing down and grinding it up. All right? So these filiform are going to be on the front 2/3 of the tongue. All right, the front 2/3. So if we draw our tongue like this, all right, the filiform papillae will be on the front 2/3. This will be the front 2/3 of the tongue. All right, and their job is just to mainly move and manipulate the food. Okay. The next type of bump, all right, is called the fungiform. All right, and literally it looks like a mushroom. That's why we call it the fungiform or fungiform papillae. All right, now they do have taste buds, not very many, just a few. All right, and so they'll be located on the tip of your tongue, all right, and on the sides. Okay, but you won't see a lot of them. Okay, all right, and then our other two, all right, we have the folate papillae. All right, these are the papillae that we see, all right, in early childhood for taste buds, all right, and they'll also be on. I'll draw the tongue again. All right, they'll also be on the sides of the tongue, but more towards the back, more towards the posterior region there. All right, and then finally our last one. This one's an easy one to remember: the valate or the circumvallate papillae. These are big, and you can actually see these pretty pronounced. If you were to open your mouth, stick out your tongue, you would see these big bumps. There's anywhere from 10 to 12 of them, all right, and they're in the shape of a V. All right, they're big, but they're so close to the back that sometimes it's hard to see, unless you open your mouth up wide enough. So how I remember is their orientation, how they're positioned on the tongue, is in the shape of a V, so they're valate. Okay, now this is where, all right, the majority of your taste buds are going to be found. Okay, but these papillae are not very numerous, all right, but their taste buds are. Okay, so when I show you this picture here, okay, you can see. Well, let me zoom in a little bit. All right, so you can see here are the valate. They're really pronounced right here, okay? And so that's where the majority of your taste buds are going to be. In the front here, all right, we've got our filiform, and they're going to be over the majority of the anterior 2/3. Again, their job is for food manipulation, toss it around. And you can see here in the diagram, all right, we don't have any taste buds. And then we've got our fungiform, and those are the ones that are going to be on the front and on the sides here, okay? And they've got a few taste buds. Then finally, our folate, which are more in the posterior lateral portion, okay? Those are going to, in early childhood, those are the ones that are going to have taste buds there, okay? But the majority, again, are going to be here in the valet. So this picture here on this slide shows you, all right, a good picture of the valet. You can see we have all these taste buds. So here's that big papilla here, all right, but you see all these taste buds, okay? Now, taste bud, all right, like I said before, when I talk about onions, they're like ogres. They have several layers, okay? So we describe our taste buds like an onion, and I wish I could turn this right side up, all right? But it's very similar to what we saw, all right? Remember when we were talking about the olfactory epithelium? Okay, we had 3 types of cells there. We had the olfactory neuron, all right, or the neuroreceptor cell there. And then we had the supporting cell, and then the basilar cell. Okay, and the basilar cell is like the baby cell, all right? So very similar here, okay? In our taste buds, we have our gustatory cells, and those are going to be the actual sensing cell, okay? Now, similar to the olfactory neuron cells, all right, they don't have a very long lifespan, okay? Where in the olfactory, all right, those cells lasted anywhere from 40 to 60 days, pretty good, decent amount of time. These guys are short-lived, 7 to 9, okay? 7 to 9. So we have the supporting cells, same role, all right? They take care of our sensory cell, the gustatory cells, and then the basal cells are just the baby cells, all right? They're going to step in, all right? They're the stem cell that's going to replace those gustatory cells every 7 to 9 days. Okay, so as we zoom in here on our picture, okay, here you can see, all right, here's our gustatory cell, and in between we have our supporting cells, and then down at the bottom near the base are the basilar cells. And our gustatory cells have, all right, these neurons attached to them, which will eventually all converge down here into the sensory neuron. All right, we're going to get into, all right, which the cranial nerves are going to be responsible, all right, for taste. And so we're going to talk about which ones, okay? So at the tip, all right, of our gustatory cells is where we're going to find our chemoreceptors, all right? And it's in a structure called the gustatory microvillus, all right? And basically what that is just dendritic endings, okay? And so they stick out through that little hole. We call that hole up there the taste pore. And that's going to be at the tongue surface there. So when you're digesting things, because actually, believe it or not, digestion occurs, it starts in the mouth. Okay, that's the first part of digestion. When you start to salivate, right, and you start to chew your food, that's the first step in digestion. So as that saliva is being produced and washing over your tongue and on the inside epithelium of your mouth, all right, well, here's a quick review for you folks. What type of epithelium, chapter 5, all right, what type of epithelium lines your cavity of the mouth or the oral cavity? You got to be specific on it. I like where you're going with the stratified. It is a stratified type of epithelium. No, not columnar. Stratified columnar is rare. Is it squamous? So it's stratified squamous, all right? But remember, we characterize stratified squamous as either keratinized or non-keratinized. So which is it? That's right. Good. No hesitation there. It's non-keratinized. Non-keratinized is the living, all right? Keratinized is the dead stuff on your skin, okay? So it's non-keratinized, stratified squamous epithelium. And if you take me in 211, you're going to get a lot of these types of questions because I'm going to revisit 210 all the time because you're going to realize how important this class is when you take 211. Okay, so, all right, in this area here, as you're producing that saliva, all right, that saliva gives a nice medium for those tastins to dissolve into, so they can interact with the chemoreceptors on the gustatory microvillus here. Okay, so these tastins, all right, dissolve in the saliva, and then they interact with the microvilli. Then they'll stimulate, all right, the sensory neurons there. Okay, so now, and you're going to see this too, and I should probably tell you this. In the rest of these these special sense, well, the rest of the chapter, all right, when we talk about sight and hearing, we're going to talk about the pathway. So where it goes from what we call the sensory organ, all right, all the way to the brain. Okay, that's going to be our sensory pathway. So we're going to start here with our gustatory pathway. Okay, so we start off with the chemoreceptors, all right, and then those chemoreceptors are going to trigger the sensory neurons. Okay, and those sensory neurons then are going to project themselves all the way to the medulla oblongata. Okay, so we go from the chemoreceptors in the gustatory cells, all right, and then they'll connect to all these sensory neurons that are going to travel to the medulla oblongata. So these sensory neurons travel in some cranial nerves. The first one is going to be facial, and the other one is going to be glossal pharyngeal. So cranial nerve number 7 is facial, all right, and that's going to be on the anterior portion of your tongue, okay, and then on the posterior portion of your tongue is going to be glossal pharyngeal. So 2 different cranial nerves, all right, anterior, all right, is for taste now, okay, not just for general sensation. General sensation is glossal pharyngeal. All right, so if you touch your tongue with your finger and you feel that, all right, on your tongue, that's glossal pharyngeal. But for taste, the anterior portion of the tongue is facial, and then the posterior portion is going to be glossal pharyngeal. Okay, so those 2 cranial nerves will project their sensory neurons back to the medulla oblongata. They'll synapse on the secondary neurons, which will then travel to the thalamus, and then from there, you know what happens. From the thalamus, all right, our tertiary neuron is then going to travel to the primary taste center or primary gustatory center, and that is going to be in the insula. That's the hidden lobe. Remember the 5 lobes of your cerebrum? That's the one that you cannot see from the outside. You have to pry the temporal lobe away, and it's deep inside. That's the insula. Okay? Oh, good. I do have a picture. All right. I'm going to come right back to these slides here. Let me just show you the picture. You know me. I love slides. I love pictures. Okay. So here you can see, all right, the anterior portion of your tongue, cranial nerve number 7, facial. Posterior is going to be cranial nerve number 9. This is for taste. Okay. So as this person is drinking their hot cocoa or coffee, they're going to stimulate those chemoreceptors here. Okay, which will then stimulate these primary sensory neurons from the taste buds. Okay, and so that's going to travel all the way back here, all right, to the medulla, specifically the nucleus solitarius. And then from there, the secondary neuron is then going to travel to the thalamus, and then the thalamus is then going to send its tertiary neurons to the cerebral cortex. And in this case, all right, that cerebral cortex is in the insula. Good? Not bad? All right, we'll come back to this picture. Which one was I on? That one. All right. All right. So you folks are familiar with taste, right? Sour, sweet, bitter. All right. Well, there's 5 types of taste sensations. Remember, what's a sensation? You guys remember that from last class? What is sensation?
I know it's conscious.
Yeah, it's a conscious perception of whatever that stimulus is. So when a stimulus occurs, when you have conscious awareness or perception of it, that's a sensation. Okay? So that's good. You threw the important word in there, conscious. Okay? So these 5 taste sensations, sweet, salty, sour, bitter, and umami or umami. Okay? So we're all familiar with the first 4. All right? Umami is a little bit different. All right? That has to do, all right, with amino acids. Okay, and that's where you get that kind of meaty flavoring too. Okay, before, when I was growing up, we only talked about 4 taste sensations. All right, umami is a relatively newer one. Okay, when I say newer one, ones that we study more. So what is important though for you folks is I want you to understand what produces these sensations. For example, sweet is going to be produced by organic compounds. Okay, a salty taste sensation will be more of metal ions. Sodium, potassium is an example. Okay, sour is going to be acidic. Bitter is going to be more basic. That's easy to remember. Basic B, bitter. All right. Alkaloids. Has anyone ever tried unsweetened chocolate, by the way? What do you think of it? Do you love it?
No.
No one loves unsweetened chocolate. In fact, I used to work at an Applebee's, this is a long time ago, and someone brought in a bar of unsweetened chocolate, which is usually made, use it for baking or whatever. And so the dare was, if you could eat that whole bar completely and not bring it back up, the guy that started the dare would give you $100. Nobody could do it. Nobody could eat the bar. That's just how bad that unsweetened chocolate was. I was impressed, but I tried. I maybe got not even a quarter of it, and I just couldn't do it. It was awful. All right, and then finally the umami, all right, that's going to be amino acids. All right, so you do want to know, all right, what compounds, what structures, what molecules will produce these taste sensations. That is important for you. Okay? All right, so last class we talked about, all right, certain structures in our special senses that are going to convert the stimuli energy, all right, into an action potential. Because remember, sound energy needs to get transformed into an action potential. All right, light energy needs to get transformed into an action potential. Well, we have to transform, all right, these chemoreceptors, whatever that stimuli is, all right, that chemical whatever, into an action potential. And remember, what did we call those things that transform the stimulus, that stimulus energy into an action potential? Do you remember? Transducers. Okay, transducers. So we're going to talk about the transduction, all right, of the gustatory cells. Okay, so for these three here, sweet, bitter, and umami, okay, those tastants are going to undergo a secondary messenger system. And if you don't remember what a secondary messenger system is, think of it like this. This is like you having a butler. I'm a messenger of some sort. I come to your front door. I knock on the door. I need to give you a message, okay? But I can't because you're in the shower or otherwise indisposed. So I give the message to the butler. The butler takes the message to you, okay? But does not allow me in the house, okay? That's how the secondary messenger system works in our cells, okay? So you have a molecule on the outside of the cell, all right, that's going to bind onto a receptor outside the cell. And what that will do is that will activate some sort of molecule. In this case, all right, we are going to have the G protein, okay? That's going to be activated, and that's somewhat easy to remember because guess what? G protein, gustatory, okay? So that secondary messenger, that G protein gets activated, all right, once the tastant binds onto, all right, the cell receptor, all right? And then what that does is, all right, that causes depolarization of the cell. And of course, when we get depolarization of the cell, we get a nerve signal, an action potential, and that's what happens here. Now, for salty and sour, we don't need a secondary messenger system, all right? We can depolarize the cell membrane directly. Part of that is because when we're talking about salty, we're dealing with sodium and potassium ions, okay? So we don't need a secondary messenger system for salty and sour. We depolarize the cell directly, okay? And when that occurs, when we depolarize those cells directly, all right, those chemoreceptor cells, all right, then we've created that action potential that's going to get produced down that primary neuron. And the primary neurons in this, all right, sensation are going to be cranial nerve 7 and cranial nerve 9. Okay, facial nerve and glossopharyngeal nerve. Those nerves house the primary neurons. Okay? So once the primary neuron gets activated through those cranial nerves, all right, they're going to wind up in, all right, the nucleus. You need to know this, solitaris. It's in the medulla oblongata. Okay? So it will then synapse onto the secondary neuron, which will go to the thalamus. But in the meantime, all right, here in the medulla, all right, we have other digestion centers there. So as that's occurring, while you're tasting things, now we are going to trigger more salivation, and you are going to begin the digestion process in your stomach. Okay? So you have visceral receptors there that are going to stimulate your stomach to start secreting, all right, hydrochloric acid, for example, especially if it's a protein. Proteins are a bear to digest. Okay, protein digestion starts in the mouth, and it pretty much goes everywhere. It goes throughout the digestion system, all right, which is, again, we'll get a chance to talk about that when we do digestion. Okay, but we have to start on proteins in the beginning because proteins are 200 plus amino acids. Anything that's greater than 200 amino acids strung together is considered a protein. But you can have amino acids that are 300, 400, excuse me, you can have proteins that are 300, 400 amino acids long. So we got to start to break those down. Also here in the medulla, all right, we have our reflex centers, all right, for coughing and sneezing, but also we've got reflex centers for gag and vomiting. Okay? I'm sure that most of us, if not all of us, have experienced something when we were tasting something and we get the gag reflex because you think it's nasty. Whoa, oh, that's nasty. I don't want to eat that. All right? God forbid if it's vomiting. All right? But it happens. Happened to my son. He hated shrimp so much, and we forced him to eat it that he gagged and threw it up. So now he does not eat shrimp. Now we've messed up his limbic system. All right, and specifically in the amygdala and the hippocampus now, because he's got that long-term memory stored in there of his experience with shrimp. He's not touching it. So, and I feel bad for him. I blame it on his mother. All right. So, from the primary neuron, all right, they synapse onto the secondary neuron, which will then go to the thalamus, and then from the thalamus, all right, it goes to the primary gustation cortex in the insula, and that's where you have that conscious sensation of taste. Okay, but not only that, all right, you can actually experience the temperature. This is a big one for people. Texture. Who's a texture eater? Yeah. So can you guys eat sushi, for example? Something. Yeah, I know. I know a lot of people.
I love sushi, but I can't handle grits.
Really?
At all.
Is it a texture thing?
Yeah, no matter how good they taste.
Really?
I can't handle them.
Okay.
I can't eat cheese.
Can you eat cheese?
No, I hate cheese.
Oh, all right, that's good and bad, you know. Yeah, I know. She doesn't. How about goat cheese, though? You ever had goat cheese? Any cheese?
I love goat cheese.
Yeah, so we'll have to blindfold you sometime and give you the taste test here. Try this. This is not cheese, I promise you. It's yogurt. I love this. All right, and so this last point here talks about, have you ever noticed, all right, how if you have a cold or if you plug your nose, and this was bad, I did this with the kids and I should never have done this, is when they were eating food that they didn't like, I'd say, just plug your nose and eat it, you know? So there is an interaction between olfaction and gustation. We've all experienced it. If you have allergies or if you got a bad cold and you get stuffy, you can't taste the food as well, all right? And it's because they share some of the pathways and there's interconnections between the two, all right? But yeah, I learned my lesson. I should not have told the kids that. Plug your nose, it's okay. All right, let's talk about vision. All right, so the first two aren't too bad. We kind of ease into olfaction and gustation. Those two are the ones that are, compared to the other sensations that we're going to go over, are relatively easy to learn. All right, vision gets a little bit more complicated. Hearing I used to think was really complicated, but for whatever reason I enjoy teaching hearing more than I do vision. But anyways, actually vision is considered to be your primary special sense. You guys ever heard that? All right, if you are blind, all right, your level of disability is much more greater than somebody that's considered to be deaf. All right, so that's why they consider it like your primary special sense. So what is vision? Okay, all right. So first of all right, when we're going to describe a stimuli, all right, it is anything that basically is going to trigger your photoreceptors, all right, to create an image of an object. Okay, some sort of stimuli. Okay, so important that you understand this. All right, the specific receptor that we use for vision are called photoreceptors, but they pick up three things. And this is important that you know this. The first one is the level or intensity of light. Okay, I should have made that slide a little bit more detailed. All right, the intensity of light. Low light, bright light. All right, like a mogwai. Okay, bright light. Yeah, there you go. All right, and you know, it's funny you bring that up. I was hiking this weekend with a buddy of mine, and we were talking about his big thing is movies, and he likes to talk about movie trivia. I don't know, some of you aren't old enough to know this, but PG-13 is a relatively new rating. All right, when I say new, it came out in the 80s. All right, and does anyone happen to know what the first PG-13 movie was? Probably not. It's called Red Dawn. Awesome movie, the original Red Dawn, not the remake. All right, I never saw the remake. Well, anyways, they said that there were some movies that prompted this new PG-13 rating, and Gremlins was considered to be one of them because that's a PG movie. PG? Pardon?
Was that Gremlins rated PG?
Yeah, it was technically PG. Yeah, it's PG. Steven Spielberg, and there's another Steven Spielberg movie we couldn't remember what it was that also helped to prompt the PG-13 rating system. But I don't know. That was his thing. He could have made it up for all I know. I don't know. I just thought I'd bring it up. Anyways, all right. Photoreceptors pick up light intensity, color, all right, and then movement. All right. Those are the three things that photoreceptors will pick up. So let's talk about our photoreceptors. Okay. These cells that are specific for what I just mentioned. Okay. So there's two types. Rods and cones, okay? So we're going to talk about some of the differences here, all right? Rods, number one, are more numerous. They're found in much more greater numbers throughout the retina, all right? And they're the night vision photoreceptors. So they will be activated and work in dim light. Cones, not so much, all right? So they're very sensitive cells. Right, which is great for us, all right, especially if you're outside in the middle of the night, all right, or you're in a dark room, all right, when you wake up in the middle of the night and you don't really feel like turning the lights on because you don't want to wake anybody else up in the house, so you have to maneuver around in the dark, all right, so you're really highly relying on the rods there, okay? So where we find the rods most commonly in the retina is going to be at the periphery, okay? So around the outside. So there's a pattern here with the rods, and the cones follow the opposite pattern. And what I mean by that is you'll have several rods. This is going to be my drawing for rods. All right. So you have a bunch of rods, and what they'll do is they'll converge, all right, onto what we call these bipolar cells. Okay. I'll make a special symbol for the bipolar cells. And then finally, all right, those bipolar cells will converge on what we call a ganglion cell. I'll put a G there. Then this ganglion cell, those axons form, all right, will eventually form your optic nerve, okay? So when we're talking about rods, they have a convergence type of phenomenon, okay? But when we talk about cones, it's a one-to-one relationship, which is ideal. Remember what we talked about? It's kind of a similar concept when we were talking about motor units. Remember when we talked about how motor units, when you had one motor neuron stimulating one muscle fiber, you would have more precision and more articulate control over that muscle fiber instead of having, all right, one motor neuron activate 1,000 muscle fibers, right? You wouldn't have as much precision. Well, in this case, it's kind of similar. All right, the cones will have a one-to-one relationship, all right, with the bipolar cell. So here's your cone cell, and then it's going to synapse onto the bipolar cell, all right, and then that bipolar cell will synapse onto the ganglion cell, and that ganglion cell will then send its axon off to the optic nerve, okay? So it's kind of that one-to-one relationship is very, very helpful, which gives you much sharper vision, okay? Even when we're dealing in a situation with bright light, because the cones operate better in bright light, all right? Our rods are going to be our night vision. And where we're going to find these cones in high concentration is going to be that in that structure that sits at the center of the macula luta. Remember that little dot structure in the center of the macula luta? That was called the fovea centralis. So when we were labeling things, we had that circle, all right, on the retina, and then in the center of it, there was this little dot. I believe in your atlas, it looks like a little yellow dot. All right, that's the fovea centralis, that dot. And then that circle there is called the macula luta. I'm not going to write that out. Okay? So cones operate better in the daylight, bright light. In this room, your cones are pretty much going gangbusters right now. All right? If I turn the lights off and waited 20 minutes, then your rods would be kicking butt. All right? They'd be doing their thing. All right? Right now, your rods aren't really doing too much. All right? Questions? Concerns? All right. So you definitely know the difference now between rods and cones, right? Feel pretty confident about that? Just think, cone, C, color, bright light. Okay, so here's a picture. We saw this in lab on Tuesday, okay? Remember, here's that choroid layer, and then you have the retinal, all this from here to here, that's the retina. And there's 3 layers of the retina, okay? You had the neural layer, all right, then you had the bipolar cell layer, and then you had the ganglion cell layer. So each of these, all right, it's like a cake, okay? This is your photoreceptor cell layer, okay? This is the bipolar cell layer. This is the ganglion cell layer, okay? And light goes this way. Okay. All right. So this is what we're looking at as we zoom in here a little bit closer. All right. You've got your rods and your cones. Okay. The outer segment, all right, is going to be embedded more here into the pigmented layer of your retina. Okay. And then the cone, or not the cone, but the cell body, all right, and the synaptic ends here, the terminals, are going to be closer towards your iris pupil area. Okay, so you can see as the light enters, it's going to hit the synaptic terminals first, travel then through the cell body, the inner segment, and then to the outer segment. Okay, in the outer segment, you have these disc-like structures. This picture here is showing how these older discs are getting phagocytized because you're making new discs. Okay, and in the discs, all right, that's where you have this structure, all right, we call that the photopigment. Okay, that's the protein, all right, that these photoreceptors have, all right, that are going to help to depolarize our cells. And we'll talk about that in a moment here. Okay? All right. So 2 things I want to point out about the rods. Okay? Poor sharpness of vision, but great for night vision. All right, we find those at the periphery of the retina, and they're more numerous than the cones. All right, the cones we'll find in the fovea centralis primarily. They work well in bright light, color, all right, and sharp vision. And there's 3 each of these cells for the cones. All right, you have 3 types, and they're specific for one of the primary colors. All right, it's either going to be red, blue, or green. Those are the primary colors. Okay, so a cone can't get all 3 colors. It can't get 2 colors. It's only one color specific. So you have blue cones, you have red cones, you have green cones. Okay, all on their own. All right. So now we know the players in the game. Let's talk about the process now of how we are going to transform that light energy, okay, into an action potential. All right, transduction, phototransduction is what we're going to undergo here. Okay, so we're going to convert that light energy into action potentials. Okay, and that's what our rods and cones are there for. Okay, so quick review: outer segment, inner segment, cell body, and then the synaptic terminals are the four parts to these photoreceptors. In the outer segment, all right, that's what's going to have the discs there. And in the discs, that's where we're going to have these photopigment molecules that are going to absorb the light, all right, and then they're going to be responsible for transduction. Okay, they're going to be responsible for triggering that depolarization, or in this, we'll talk about depolarization and hyperpolarization of these cells. Right? Inner segment has all the organelles to keep the cell alive. Okay, cell body has the nucleus, but importantly, at the synaptic terminals, that's where we have the neurotransmitter glutamate. You want to know that. Okay, glutamate is going to be, all right, one of the neurotransmitters that we will discuss by name, all right, that this, in this case, is going to be specific for the visual system. Okay? And we'll talk about its role, all right, but it is the neurotransmitter that's going to play a role here. All right. So I mentioned that we have these photopigment molecules, and I want to get into specific about them because you need to understand, because we're going to talk about the bleaching effect. All right, and the bleaching effect is basically you sitting in a movie theater, all right, or here, better yet, you're lying in bed, all right, and you've been lying in bed, you're sleeping, you wake up, you're like, I'm gonna go to the bathroom. So you get up to go to the bathroom, you flip the lights on. And you know how bright those lights get, all right? So you go and do your business, you go back and lay back down in your room, all right? But yet the room, to you, if your eyes are open, will not remain as dark as it was when you immediately woke up, all right? Because the rods have undergone this phenomenon called the bleaching effect, which we'll talk about, all right? But that has to do with these photopigments here, okay? So let's talk about what a photopigment is. One, it's a light-absorbing molecule, okay? It's going to be found in the outer segment there, all right, of the rods and cones, and those discs there. And it's made up of two important components. The first one is called opsin, right? And that's it's the protein that is attached to this light-absorbing retinol. Now, retinol comes from vitamin A, and that's why it's important, and where they've encouraged, the joke was, hey, eat your carrots, because carrots are high in vitamin A, and also beta-carotene. If you eat enough carrots, your skin will turn orange. True story, all right? But it has vitamin A, and with vitamin A, it helps to contribute to the retinol here, okay? So depending on which photoreceptor we're talking about will determine what type of photopigment that we're going to be discussing, all right? So when we talk about the cones, all right, for example, all right, we have different opsins in each cone because those different opsins are going to be responsible for transforming specific wavelengths into action potential or nerve energy, okay? So a blue cone, and I'm going to come right back to the slide here, okay? A blue cone, all right, is only going to be activated when, all right, this 420 wavelength hits it, all right? If you hit that blue cone with a 550 wavelength or 520 wavelength, it's not going to stimulate it, it's not going to activate it because it doesn't have the proper opsin protein to be stimulated, okay? So depending on what cone you're dealing with, all right, it'll have a specific opsin for a specific wavelength. That's the point, okay? So and then don't forget rods too, even though they don't have, they don't see color, but they still have, all right, their opsin, okay, is specific right, for a specific wavelength also, okay? So when we're in with the rods, easy to remember, all right, the opsin molecule is called rhodopsin, okay? And then we're talking about cones, all right, we call those photopsin, all right? It just varies, red photopsin, blue photopsin, green photopsin, okay? And all the photopsins, all right, is going to be based on whatever wavelength, all right, will activate it. All right, so if you look here on this picture here, all right, the shortest wavelength is going to be for the blue wavelength, okay? And then for red, okay, that'll be the largest wavelength. Okay, rods are about 500, green's going to be about 531, all right? It's just different wavelengths, all right, will stimulate those opsin molecules. So put that into memory, all right? Rods are going to be rhodopsin, okay, and then the cones will have photopsin, and they all vary depending on the specific wavelength that they're designed for. Is that good? Did you guys kind of understand where I'm coming from with that? All right, it's going to be specific. Which brings me to color blindness. Does anyone know anybody that's colorblind? You know, or you do? Are you colorblind?
I would like to.
Okay, okay, yeah. Yeah, my father-in-law is colorblind, which is crazy to me because he likes to go hunting. And I'm like, if you don't kill the deer, all right, and it runs away, how do you follow the blood trail? You can't see it. It's red, you know? He swears that he can do it. But anyways, when I was younger, you don't see it too much anymore, but when I was younger in some of the smaller towns, right, when you drive up to a traffic, to a four-way, you know, intersection with the traffic signals there, all right, it's got the three lights there, okay? Red, yellow, green, okay? Well, we all know, for the most part, I hope you know, all right, red's on the top, yellow's in the middle, green's on the bottom. If you ask someone randomly, they might not know that. You'd be surprised how many people don't know that, all right? But in some of the smaller towns and older towns, they used to have the traffic signals on the side. They were horizontal instead of vertical, all right? A colorblind person knows the signals, all right, red's on top, green's on the bottom. But in those smaller towns, when they used to put them on the side, a colorblind person couldn't distinguish which was which, you know, because there wasn't a normal pattern to that. So that's one of the reasons why they stopped doing that, bless you, all right? But most of the time, colorblind people, all right, they can't distinguish, all right, between, it's usually brown and reds, or no, green and reds, excuse me. With my father-in-law, it's all, a lot of things just look like brown to him, he said. He said it just looks brown, which is interesting to me because his outfits are pretty well coordinated, but I think his wife dresses him. I don't know. But with color blindness, it is an X-linked recessive condition, which makes it more common for men because we only have one X chromosome. Now, you ladies, in order for you to be colorblind, we got to knock out both X's, which is harder to do. Okay, but for us men, you knock out the X chromosome, we're most likely going to be colorblind. It could be, yeah.
A lot of like number one shirt goes with pants number one.
Oh, really?
Yeah, like his wife.
Clever.
Dress himself based on the numbers.
If she gets mad at him, she gives him the wrong color shirt. You make you look like a jerk today, honey. It's like, I know with blind people, they'll fold their bills in a certain configuration so they know if they're spending a $5 bill, $20 bill, $10 bill, which I think is pretty clever. All right, we're getting close to the bleaching effect here, but I want to jump into, all right, some more phototransduction here, okay? So staying on that concept of turning that light energy into, all right, an actual potential, we're going to talk about what happens when you're in a dark room, like what I was mentioning. So that same example, all right, you're going to get up to go to the bathroom, okay? So when you're going to get up in the middle of the night to go to the bathroom, you know, you've been sleeping for a while, all your rods, okay, that's the night vision molecule, okay? They're going to have, and I'm going to show you a picture here, all right, the rhodopsin, all right, is going to be in what we call the cis-retinal configuration, okay? So I'm going to show you real quick. I'm going to go back and forth on these, okay? All right, so here's your photomolecule here, all right? This is the light-absorbing molecule. And the rhodopsin, here's the opsin protein, this dumbbell-looking thing, And then you've got the cis-retinal, that's the light absorbing molecule here. All right, it's in this configuration. Essentially, it's bent. Right, the cis-retinal, all right, photo-absorbing molecule is bent. Okay, so when you're sleeping at night, you're lying there, all right, that molecule is in that configuration. So when you open your eyes in a dark room, you're able to, see better. Okay. So now, when you go into the bathroom and you flip the light on, all right, that cis-retinal then turns into the trans-retinal. It gets reconfigured. So basically, you had like something that was bent, and now we straighten it out for you. Okay. But regardless, that process, all right, that changing of that molecule causes, all right, the trans-retinal to pop off the rhodopsin, which basically inactivates it. So it doesn't work, okay? So here you go. You go into the bathroom, you flip on the light here, okay? That cis-retinal molecule undergoes a configuration change, and it transforms into the trans-retinal, okay? So it basically straightens out, right? When that occurs, that trans-retinal pops off of that opsin protein there, okay? And so now, that process is called the bleaching reaction, okay? The bleaching reaction. So when that pops off, that is going to cause where your rods, all right, aren't going to be able to operate for roughly about 20 minutes, right? Because it takes a long time. Now the reason why, all right, so let me go back to our story. So you've done your business in the bathroom, you go back in and you lay down. So this is what happens after the bleaching. So you're laying down there and your night vision just won't be the same. It won't be as good, okay? And that's because it takes time. We have to take the opsin protein, and we've got to take, all right, our light absorbing molecule, the retinal, which is trans-retinal configuration now, and we got to stick it back on there, okay? But we've got to convert the trans-retinal back into cis-retinal. All right, so we have to basically rebuild, all right, our trans-retinal and convert it back into the cis-retinal. Well, guess what? That takes a little bit of energy, all right, in the form of ATP. When that happens, then we can transport the cis-retinal, all right, back onto the opsin. But guess what? All right, that we have to take that trans-retinal out of the photoreceptive layer, and we have to transport it all the way into the pigmented layer. Where's the pigmented layer? Here. So we have to take that molecule, all right, out of these discs, because that's where this molecule is, all right, that photopigment is here in the discs, all right. We got to take it out of the disc, back in here into the pigmented layer. So it comes out of the cell, then we have to reconfigure it, bend it again, all right, using ATP, bend it back into that cis-retinal configuration, all right, and then we got to take that, all right, and then stick it back onto the opsin here, okay, and then we can reconfigure and get our rhodopsin back, okay, so that takes time. It also takes ATP, okay, and that roughly takes about 20 minutes, all right. And so that's why, if you've ever noticed, all right, not that you probably stay awake that long after you use the bathroom in the middle of the night, but if you were to go outside, for, let's say, maybe you want to look at the stars, you walk out of your house and there's no moon, there's no full moon, in fact, there's nothing but stars, it will take you about 20 minutes to get your night vision in full effect so you can really appreciate the stars if you want to go and look at the stars there, okay? So it does take a while for that to happen, and that's why, all right, like I said earlier, when we're sitting in this room, our rods aren't going to be working because this is actually technically considered bright light. I mean, not as bright as someone flashed a flashlight in your face, but all right, it is considered bright light. Now, this process can occur in the cones too, but it happens much faster. All right, much faster. And the light that's needed for the bleaching effect in cones is, now that will be a flashlight, because you've seen, like if you get a bright light flashed in your face, all right, and you'll see like spots, all right? Now we've affected the cones, all right, and that's usually from an intense light. But they won't stay bleached as long, all right? Our rods stay bleached for about 20 minutes. Cones, not as long. So that brings me to both, what do you consider bright versus dim lights? Okay, great question, great question. Bright lights, perfect example. When you're driving at home tonight and you see a car coming at you and they've got their high beams on, well, there's another word that I want to call that, but that's bright lights, all right, that's bright lights, okay? So dim light would be movie theater kind of atmosphere, okay? If you've got a dimmer switch on your light in your room or dining room or whatever, you turn it down to the lowest setting, that's a dim light, okay? Candlelight's a dim light, okay? Walking outside in your neighborhood, even though people have their front porch lights on, that's dim light. So that brings me to dark and light adaptation. All right. So this is what I was talking about. All right. The dark adaptation. All right. So when we have our bleached rods here, about 20 to 30 minutes. All right. To return back to our normal vision. All right. Light adaptation. Okay. This is just the opposite. When we go from a low light setting to a bright setting. Now we have the help of our pupils because they'll constrict and let less light in. All right. But still, okay, the cones will still feel the blunt effect of that. But the nice thing is that they regenerate much faster than the rods. And so it's about anywhere from 5 to 10 minutes. Okay, so just think of those spots that you're seeing. They don't stick around too long. Okay, those bright light spots. All right, this brings me into, all right, now we talked about what happens, okay, in the photoreceptors. Okay, but now remember, we've got the photoreceptor cells are going to get activated. Okay, but then we've got two other layers, all right, that they actually have to communicate with: the bipolar cell layer, all right, and then our ganglion cell layer. Okay, so I talked about what happened. Where am I going? Here. All right, I talked about what happened in this layer first. All right, this is your photoreceptor layer. All right, but now we've got to talk about these bipolar cells here, and then our ganglion cells, because the ganglion cells, all right, if they are stimulated to create an action potential, this layer right here, this is the inside of the retina. That's where all the axons are for these ganglion cells, which are going to transmit those action potentials to the optic disc. All right, and then eventually back on the optic nerve. Okay. Where am I? No. Yeah. Okay. So let's talk about what happens in the dark. All right. So going back to that same example. Okay. You're laying in a dark room, let's say. You don't even have to go to the bathroom. You're just laying in the dark room. All right. So with the rods, okay, when you're laying there in this dark room, in that dim light, all right, they are going to be depolarized. So that means, all right, the membrane potential is going to be at that negative 40 millivolts, okay? I'm not going to get into too much of the details here, but this compound here, the cyclic GMP, all right, guanosine monophosphate, this compound is being created, and what it does is it opens up your positive ion or your cation channels, and that allows both sodium and calcium to enter into the cell. And you know, when positively charged ions enter into the cell, they make the inside of the cell less negative, all right, or more positive. So they'll depolarize the cell. So as they do that, all right, we refer to this as the dark current. Okay? It's kind of like the dark web. Okay? We don't talk about the dark web. All right? I read an article about this guy. I know it's crazy. About this guy. Well, actually, the article is about his girlfriend. Apparently, this guy started this website on the dark web. And on this website, you could hire people. You could buy drugs, prostitutes. But the part that blew my mind was that you could hire people to assassinate other people. on this guy's website. And I guess the FBI was hunting. Has anyone heard this story? Have you heard this? Yeah, it's nuts. I mean, this guy he looked like any one of us, you know? I mean, he was a college student at one point, just a regular dude, but he spent a lot of time in his room. And the girlfriend, the article was talking about when they interviewed the girlfriend, she says, like, come on, let's go out and do stuff. And he's always in the room, and she didn't really know what he was doing in there. And then they kind of broke up, and they were trying to reconnect, but then he got busted by the FBI. But it was nuts. I mean, I'm thinking this guy was doing all this stuff unbeknownst to anybody. All right, anyways, so here's what's going on. All right, as we depolarize the cell, all right, what this causes, all right, it causes these, remember, at the synaptic knobs in our normal neuron, we have our voltage-gated calcium channels. So as we're depolarizing, all right, the neuron, all right, these voltage-gated calcium channels are going to open up. And guess what? They're going to release the neurotransmitter. In this case, our neurotransmitter is glutamate. Here's the crazy part about glutamate, all right, in the eye, okay? This is going to hyperpolarize, all right, the bipolar cells. Now, when it hyperpolarizes the bipolar cells, as you know, it makes it much more difficult to depolarize. All right? So what happens then is, since they're hyperpolarized, they cannot be activated. All right? So they will not activate the ganglion cells. Ganglion cells do nothing. So I'm going to show you a quick picture just to kind of make a little bit of sense of what I'm talking about here. All right, this is in dark light, okay, in the dark with our rods, okay? So here's our photoreceptor cell, okay? So what I mentioned about the transduction is all occurring. So basically what happens is, all right, we depolarize the cell, okay, and sodium and calcium enter in, and they depolarize the cell, and so it creates, all right, a graded potential, all right, that goes down towards the cell body here, and then eventually down here, all right, to the synaptic terminals, which triggers the opening of our voltage-gated calcium channels. So, like we learned back in chapter 12, all right, those voltage-gated calcium channels, all right, allow calcium to enter into the cell. Okay, it gloms onto the synaptic vesicles, causes the synaptic vesicles to release the neurotransmitter. In this case, it's glutamate, right? So our neurotransmitter then, okay, travels across the synaptic cleft, and what it does to the bipolar cell is it hyperpolarizes it, thereby preventing the opening of voltage-gated calcium channels on the bipolar cell from opening up, so therefore the ganglion cell does nothing. Okay? No nerve signal gets sent from the ganglion cell. Okay? And this is what happens in the dark with our rods. Okay? So now, what happens when you're exposed to light? Okay? Right here. What's happening in this room? Okay? I'll tell you what happens, all right? So in this situation, excuse me, in this situation, the light, excuse me, the light will hyperpolarize the rods. Okay? So now we'll go through what happens. All right. So we talked about what happens with rhodopsin. Splits the rhodopsin and whatnot. But in that process, we have activated our secondary messenger system, the G protein. Okay? And when that occurs, all right, that G protein there, all right, activates or breaks down our cyclic GMP, all right, which, excuse me, which causes closure of those of those sodium and potassium, excuse me, sodium and calcium channels. So when sodium and calcium are prevented from entering into the cell, all right, we're not depolarizing the cell, but in this case, we are hyperpolarizing the cell. And so since that photoreceptor cell is hyperpolarized, okay, we're not generating those graded potentials, all right, and so we're not creating an electrical signal, so that maintains these voltage-gated calcium channels to remain closed. And because of that, those those cells are not releasing that glutamate, okay? Because we saw glutamate will hyperpolarize the bipolar cells. So now we're no longer hyperpolarizing the bipolar cells, all right? So since we're not hyperpolarizing the bipolar cells, now the bipolar cells can release their own glutamate onto the ganglion cell. And that glutamate, all right, neurotransmitter excites the ganglion cell, and that ganglion cell will then produce an action potential, all right, and it'll send its action potential or nerve signal, all right, down the optic nerve, cranial nerve number 2. Let's look at that in the picture here. Oops. All right. All right, so here's your rod. Flip the light on, okay? And what we've done now, all right, because we no longer have that cyclic GMP being produced, it causes, all right, our sodium and calcium channels from opening up. They stay closed, okay? And because of that, we are no longer producing graded potentials down along the photoreceptor here, and therefore we're not opening up these voltage-gated calcium channels to release the glutamate. Since we're not releasing glutamate on the bipolar cell, that allows the bipolar cell, it's no longer being inhibited, okay? It depolarizes, okay? And it releases, all right, its own glutamate onto the ganglion cell, which actually stimulates and activates the ganglion cell, all right, to send out its nerve signal, all right, out to the optic nerve, okay? This is probably one of the more complicated aspects of this chapter. So if you have a pretty decent understanding, there's really no exciting way to explain it to you guys, and I apologize. I try to do as best I can. This is one of those things where you just kind of memorize some of the aspects of it. If you know, all right, this picture, okay, you'll do pretty well, okay? All right, let's talk about, what time is it? Oh, good. We're doing well. You guys are doing well. You're hanging in there. Let's talk about light refraction, the bending of light. This concept is important because for any of you that are wearing contacts or glasses, such as myself, we are all taking advantage of light refraction, okay? And basically, when you think of light refraction, what we're doing is bending light, all right, to a central point. Hopefully, that central point will be the fovea centralis, where all those cones are located for sharp vision, okay? So we're gonna bend the light and focus it, much like if you've ever taken a magnifying glass. God, I hope you've never burnt ants with it. But when you take a magnifying glass with the sunlight and you burn maybe a leaf with it, that's because you're focusing those light rays on a central point. What we want to do with light energy rays is focus them on a central point on the retina. Hopefully the fovea centralis. That's where our sharpest vision is going to be. So this picture is showing us, all right, the actual concept of refraction, okay? We're seeing how the spoon, all right, when it's put into water and you compare how light is reflecting off the spoon in the air versus how it is in the water, okay? It's just showing us that the water is bending the light rays, okay? And that's why it looks like the spoon is broken, okay? So one of the things you need to understand is, one, light when it travels through different types of media, all right, air versus water versus a solid, like a prism, all right, those different media will bend those light rays differently, okay? So the lens, all right, either in your glasses or the lens of your eye is going to be responsible for the majority of the light rays that get bent. Your cornea bends light a little bit. The vitreous and aqueous humor, they bend light a little bit, okay? But mainly the bending of light or the refraction of light is going to be solely responsible by your lens there, okay? We'll talk about that. All right, let's talk about what happens when you are going to be looking at different objects that are close by and then objects that are far away. All right, So the first one is called the near response. All right, this is when you're looking at something close by. All right, if you're looking, well, it depends on how far you are, but if you're looking at your computer screen, or even that screen here on the front of the room here, okay? Basically what's going to happen is as you're looking at something relatively close by, your medial recti muscles, all right, are going to cause your eyes to converge, okay? It's also going to cause accommodation. We'll get to that in a second, what accommodation. We talked about that on Tuesday. And then your pupil will actually constrict a little bit. And the reason why your pupil constricts a little bit is to make sure that it's not letting in too much extra light. Because if too much light gets in, it'll start bouncing around in there and can distort the image. It can be a little bit fuzzy. It just won't be as focused. Okay? So the first part, all right, when we talk about convergence, as your eyes move medial to one another, that's going to be your medial rectus. And ideally, what you're trying to do is focus, all right, and get the image. Because remember, your right eye is getting an image, and your left eye is getting an image. Okay? And what your brain does is pretty much overlap those images onto one another to give you one depth perception. All right? That actually is called binocular vision. Okay? So each eye is getting its own image, and then your brain puts it together and gives it depth and whatnot. Okay? So ideally, what you want to do is get those images on both of the fovea centralis. Okay? If you have a weakened eye muscle, okay, either or, you can get this condition called diplopia, double vision, which happens quite frequently in people that drink. All right, well, they get relaxed, but that's not the reason why they get double vision. All right, but if you do have a weakened medial rectus muscle, right, then you can't get that convergence of the eye properly, and so you will not be able to get, all right, that sharp vision on the fovea centralis there. All right, we talked about accommodation, but let me just kind of review it real quick. Accommodation is when you get an alteration in the shape of the lens. Okay, in this case, the lens is going to be thickened. It's not going to be thin. All right, it's going to get nice and fat like that. And what causes that is going to be the ciliary muscle will contract. But remember what I said, all right? When the ciliary muscle contracts, it causes slack to occur in the suspensory ligaments. Those are those ligaments that attach onto, all right, the lens, okay, that we saw. And so when those suspensory ligaments are slackened, you do have some elastic fibers in the lens, and it I won't say shrinks, but it assumes this kind of configuration, right? It becomes thick, okay? And when it becomes thick, all right, we actually increase the amount of light bending or refraction that occurs, all right? So then finally, we have constriction of the pupil, and that's due to the sphincter pupillae muscle, which is under what division of the autonomic nervous system, all right, activates the sphincter pupillae muscle? The division, remember the two divisions of the autonomic nervous system, fight or flight, rest and digest? Which one do you think it is? Which one? The parasympathetic. Yeah. Specifically, now, if you can wow me, hit me if you know which one specifically, which cranial nerve. Remember, there's 4 in the parasympathetic. There's 3 oculomotor, 7 is facial, all right? 9 is glossopharyngeal, and vagus is 10. Yeah, I like it. I'll go with that, I think. I'm just kidding. All right. Okay. So what happens here again, as your pupil becomes smaller, all right, we'll get less light to pass through the lens. That's going to decrease the blurry, any type of blurriness there. Okay. All right. And now, if you were to look at something far away, like the mountains far away, right? We will not have any type of convergence that occurs. Accommodation will not exist, so the lens lengthens because the ciliary muscles relax, but the suspensory ligaments become tightened. Okay? You'll get less refraction, right? But in this case, you will get dilation. Okay? So more light can get in there. So here's a quick picture here, all right, of near and far vision. Okay? So you can see here, all right, this top one here for distant vision, we're looking in the mountains here, all right, the Appalachian Mountains. All right, the lens has become thinner, all right, the ciliary muscle is relaxed, but the suspensory ligaments, sorry, hold on, let me zoom in. All right, the suspensory ligaments, it's hard to see, have become taut. They're tight, they're straight. Okay, so we get less bending, all right, or less refraction, okay, as that light image passes through and gets reflected onto the retina here. Okay, now you can see here that that image gets turned upside down and all that. It's true. I'm not going to get into all that. Then your brain flips it back again. Okay, so there's a lot that happens. The visual pathway is awesome. We're going to go through just the basics of it, but I could do some fun stuff with that. But time is of the essence. All right, accommodation. All right, now you glance at your wristwatch. Okay, after looking at the mountains. Okay, so the pupil starts to constrict. All right, letting in less light. All right, your lens becomes thick because the ciliary muscles contract, and those ciliary muscles, when they contract, the suspensory ligaments become slackened, so the lens kind of rebounds back to this shape, causing more bending of the lights, more refraction. All right, and so you're going to see, all right, more focusing on that close object there. Right, and also we have convergence. The eyes converge. Okay. Now, as you get older, and actually, I think we have time. We might. When you get older, all right, the lens has a harder time with accommodation, hence reading glasses. Okay, and we'll talk about that in a second. Cataracts. Anybody here have a cataract? Going once, going twice? Good, awesome. All right, in case you were wondering what a cataract was, all right, in the lens, all right, you get these what we call opacities. Okay, and I was talking to the other section the other night. Those aren't the same as floaters. Does anyone know what I'm talking about with floaters? Yeah, sometimes you'll just be, if you kind of, I can't do it right now, but sometimes I'll see something move across my field of vision. All right, and that's not a cataract. That's just, yeah, you'll get them. In fact, you get them when you get older. You get more of them as you get older because you start to get calcium deposits and other structures in the vitreous and aqueous humors that are floating, that are in your eye. All right, not the same thing. These occur in the lens. All right, those are in the lens, and it happens as we get older. Okay, so it will affect your clarity of vision there. All right, so one of the techniques that they use now that they can break them up a little bit. I think there's I can't remember the name of it, in which they can use a laser. It used to be a big deal to get a cataract surgery. I mean, I'm talking about like 20, 30 years ago. Now, I mean, my patient's like, I'm having cataract surgery on Thursday. I'm like, all right, well, I'll see you in 2 weeks. They're like, no, I'll come in on Friday. I'm like, all right. So it's not that big of a deal. And especially when they use the phacoemulsification, when they're just using sound waves that just bust up, all right, those opacities there, and they just pluck them right out of the lens. It's cool. It's amazing what they can do. Cool stuff. All right, let me just wrap up here real quick with visual impairments, because I'm sure we're all dying to know, especially those that wear corrective lenses or whatnot. All right. So if you're normal, all right, and I don't know how many of us in here are, all right, but you have that 20/20 vision, or even better if you have 25/20 vision or 30/20 vision, okay? But that's called I always have a problem pronouncing this. Emotria, all right? That's normal vision, all right? Both eyes are able to focus perfectly, all right? All right, if you are farsighted, okay, this is, I'm nearsighted, so I'm myopic. You're both? You are? Wait, really?
I'm old.
Oh, well then, yeah, then you probably have presbyopia. See, I've got presbyopia, and well, I've got three things. I got astigmatism, I'm presbyotic, and then I'm myopic. All right, so I have problems, and you. So it's interesting because so you were told that you're hyperopic, and myopic. Oh, okay.
But I'm only, I think myopic because I have a wrinkle in my cornea
Okay.
that's like
Oh,
directly
okay.
in front of my lens.
I got you. Hold on, what you say? Heather, you said I'm farsighted in one eye, nearsighted in the other with astigmatism. Okay, all right. Yeah, and that's and that's what I tell patients all the time. You're allowed to have more than one problem. All right, that's just one of the things. But let me jump into this, and then we can talk more. All right. So if you're a farsighted individual, all right, your eyeball is going to be considered to be too short, and so what happens is, all right, the image. I'll show you the picture here. The image of whatever you're looking at, all right, is going to occur behind the retina, all right, where it's supposed to focus. All right, it needs to focus here on the fovea centralis, all right, but that image comes to a focus behind because your eye is too short. All right, so hence in that type of situation, when they give you corrective lenses, they're going to give you a convex lens. All right, that's a lens that kind of that's an awful drawing. It looks like that. Okay, and so that will bend and cause. That's why if you've ever gone, well, if you're myopic and all that, then you've gone to an eye doctor, and that's when they do the lens thing. You're looking through that machine there, like, all right, can you see better now, or do you see better now? And they change the lenses, and that's what they're doing. They're just trying to find the right lens that bends the light perfectly for you. All right, so if you're myopic, all right, that's me. I'm nearsighted. My eyeball is too long, so when all those light rays converge, all right, they converge in front of the retina. Okay, and so my lenses are concave, so they're going to look more like this. Okay, so that helps to get the light to focus here on the retina. So showing myopic individuals, all right, without my glasses, they come to a focus in front of my retina. With my glasses, they focus right here on the retina. Okay, if you don't know if you're myopic or hyopic, find out. It's helpful. And then, if you're old, like me, you can be presbyopic, okay? And that's when my lens has issues changing shape, okay? It has more difficulty becoming spherical like it should, for accommodation, all right, because it's lost that elasticity, all right? LASIK has been something that's been one of those techniques that's been able to help folks, all right? But I figured, you know what, I'm just going to use some glasses. Works good enough for me. And then stigmatism, I had an ophthalmologist once tell me that with astigmatism, instead of your eye being circular, it's more like a football, like that. All right. But again, that is, and because it's abnormally shaped, it causes a problem with those refractive surfaces there, and it causes that inability to focus appropriately. All right, but I don't think I have a picture for that one. No, I don't. I don't. All right, let's take a break. I know your brains are probably full, and I covered everything that I wanted to. And then when we come back next week, all right, on Tuesday, we'll finish up the chapter, and we'll talk about the visual pathways. And then we get to do vision and equilibrium.